everyone, and welcome to PPMD's webinar, Resiliency and Results, What We Learn When Trials Are Terminated. I'm Eric Camino. I'm PPMD's Director of Research and Clinical Innovation. I think as everyone in this community knows, clinical trials are one of the biggest sources of hope and inspiration in our community. Duchenne is fortunate that we have such a robust pipeline of potential therapies in development, but as we are all aware, not all of those investigational products are realized as approved therapies. And when trials are terminated, it can be devastating to families. But it's important to recognize that termination of a clinical trial does not equate to failure. That's in part why we're referring to this as resiliency and results. All of that time spent by families and individuals with Duchenne and Becker in clinical trials continues to yield new learnings that can help advance the field of Duchenne. And today we're going to hear about some of those lessons that have continued to provide benefit to this community even after a trial has stopped. Next slide, please. I'm joined today by a number of great speakers from Pfizer, Roche, Genentech, and Wave Life Sciences, along with Pat Furlong, the President and CEO of PPMD, and we're going to hear from each one of these sponsors about one of their clinical trials and the lessons that they've learned from that despite the trial stopping. Next slide, please. So just a little bit of an overview for how this is going to unfold as we have a number of speakers. So we're going to talk a little bit about kind of the goals here, some expectations in drug development, and then we'll go through with each of the sponsors some of the lessons that they've learned before opening up to a community question and answer. Next slide, please. Just to start, and I kind of had that little bullet point about sharing information, and I think that's a really important aspect of this is that, you know, we need these opportunities for the community to connect with sponsors to connect with the community and with each other. So that way, all of these lessons that we're going to see here today can be shared between us, that we can get input so that the sponsors can learn from each other, and so that your input on clinical trials can be heard and potentially be incorporated into the future design of trials. And I just wanted to put some expectations that out there that we sometimes don't talk about when we think about drug development before getting into the lessons learned. And that's, you know, the first one is that drug development is very challenging. And I do think that's something this community is very aware of. You know, the statistic is kind of on the average that 1 in 10 investigational products become approved. And the reason why so many of those products don't make it to that approval stage is for a myriad of reasons. And that can be challenges with the safety profile of an investigational product, its target engagement, the efficacy that we see once we're testing in humans, and even challenges with, you know, recruitment and patient participation. And so these are challenges that we've dealt with in the past, and that we will continue to deal with. But it's important that even in those instances when we don't see a product make it all the way to approval, that we are still learning from that and sharing that with the community, which is, I think, what you're going to hear today. And part of this is because investigational products harbor unknowns. This is, you know, the reality of drug development. These investigational products are developed on sound science and preclinical experiments, but ultimately they need to be tested in humans, and that translatability is not perfect. Our preclinical models are not one-to-one representations of humans. And for anyone who's interested in maybe learning a little bit more on translatability, we have a wonderful presentation from Dr. Erasmus Roos on our virtual conference that kind of explains some of those preclinical models and how they're used in the drug development process. But, you know, ultimately what we're talking about today is that resiliency and hope, and that, again, I'm stressing that termination of a clinical trial is not the end. That does not equate to failure. There's still a lot that can be learned, and it's important that we have resiliency in the face of termination, that we continue to create an environment that encourages sponsors to develop and test compounds that can become potential therapies. And many of you in this community have contributed to that, either by participating in clinical trials, in natural history studies, in your work of advocacy with Congress in finding ways to allocate resources to further our research of Duchenne. And all of that work goes into expanding our knowledge, and in that expansion of knowledge, we have better chances to develop promising therapies to help treat and cure this disease. And then lastly, before I hand this over, I think it's important to recognize that we are having quite a bit of success in Duchenne, and it is very promising. We now have 4 approved therapies. We have, an additional new drug application filed recently. We have a number of trials that are in their phase 3 studies. We are on the cutting edge of gene therapy trials, and all of this, in the rare disease space. It's really astounding, and I think holds a lot of promise for the future. So, to begin in this series of lessons learned, we are going to be joined first by members of the Pfizer team to talk about their clinical trial for Dehydrogenase. And some of the people on the line may not have been in the community or more recently joined. So for those who are unaware, Dehydrogenase is a monoclonal anti-myostatin antibody, and the myostatin protein is an inhibitor of muscle growth, and so this antibody was targeted to bind that myostatin protein and block its activity. And so the phase 2 Dehydrogenase trial was terminated in August of 2018, but as you will hear, the Pfizer team has continued to learn and share their findings with the Duchenne community, and we're grateful for that. So joining us today, we have Dr. Lawrence Charnas, who's the Executive Director of the Rare Disease Research Unit. We have Shannon Marafino, the Senior Director of the Rare Disease Research Unit, and Dr. Sarah Sherlock, who's the Director of Early Clinical Development. And so with that, I will hand it over to the Pfizer team.
Thank you very much, Eric. This is Shannon Marafino. I'm going to walk you through these slides today with the help of Sarah. So if you could just go to the next slide. So we just provide this disclaimer here that this information is being presented and it's publicly available in response to PPMD's request for communication on this compound. And these study results have been posted on clinicaltrial.gov under this NCT number. Next slide. So just quickly, the 3 topics that we wanted to cover briefly today. First are what does a phase 2 study success mean? Then we wanted to talk a little bit about the Dehydrogenase study specifically and what we, Pfizer and the DMD community, have learned by conducting this clinical trial. And then finally, we wanted to talk a little bit about data sharing and reporting. And next slide. So one of the questions is why do phase 2 studies fail to meet all their objectives? And we really want to emphasize all here. There are many objectives that go into developing a clinical trial, but primarily when we design a clinical trial, it's done to test what's called a hypothesis. And a hypothesis really means that we're looking for usually the efficacy of the drug. So up front, there's usually a stated hypothesis that is provided, and we then apply a statistical method to measure whether we can observe what is stated as the hypothesis. And I just gave an example here, really based again on our Dehydrogenase drug. And the example would be that our hypothesis was that drug X would slow the functional decline in Duchenne patients greater than the current standard of care. Now, as Eric was saying, when we talk about the outcomes of clinical trials, it's really important the language that we use. And in particular, sometimes we hear when a study is read out as negative, meaning the drug was not able to slow the functional decline in DMD patients compared to standard of care. That might be reported as a failure. And really what that implies to a scientist is that the study couldn't test the stated hypothesis, and therefore we can't learn from the conducted study. And it's really important that we use the right language when we talk about a study reading out that, whether it was positive or negative. But when we use the word failure, it really does, again, suggest that we can't, really understand or learn from that clinical trial. So it is unfortunate that despite having detailed plans and best efforts, we do often see phase 2 studies, especially around new drugs, remain quite low. And there's many, many reasons for that. These are just a few of the reasons listed. Sometimes, as Eric was saying, it comes down to poor translation. The animal models, for whatever reason, there's many reasons, don't translate, well when it gets into a patient. Also, the pharmacology or the effect of the drug may not be relevant for the patient, or the disease or the target really isn't clearly relevant. And then we also have, potential issues around disease characteristics. Patients come in, they have different symptoms, and they have different progression across the various stages of the disease. So that can add a lot of variability to what you're trying to measure. And then finally, understanding, the outcome measures in any disease are important. And we know in Duchenne that these have continued to evolve over the years as clinical trials have been conducted. And then sometimes these outcome measures take a long time to observe. So that can also be a challenge. If you can go to the next slide. So one of the things that we try to do to bring more success to our phase 2 programs is really to characterize the properties of a drug. And by doing that, we actually have what we call the 3 pillars, and this has been published by Morgan et al. Around different drug characteristics that help us to achieve a more successful phase 2 program. And what I've tried to do here is describe the 3 pillars, and then I wanted to give you an application to how those apply specifically to domogrozumab and what we were challenged with during our drug development for this product. The first pillar that we have is looking for what they call PK exposure. So when we look at PK, we're actually trying to measure if we can measure that drug at the target where it's intended to go. So in the example, optimally, we should be able to, for domogrozumab, we should have been able to measure that drug within the muscle. That's the target compartment, where that drug is supposed to go. The actual circumstances, though, for DMD is that it's really not possible to measure the drug in the muscle compartment. One, because the muscle, goes throughout the entire body. So, really it would be impossible for you to measure every, muscle in the body to see if the drug was available. However, what we can do as a substitute is measure the drug circulation in the blood. And that's actually a pretty good surrogate or a substitute for it being able to see if we are able to see that drug in circulation. The second pillar that we look at is if the drug is actually able to bind to its target. So this is the anti-myostatin. It's a monoclonal antibody, and its job is to inhibit myostatin, which is a protein, which is a negative regulator of muscle growth. So ideally, the question is the binding in the target in the muscle compartment sufficient? So can we bind up all that protein in the muscle with our antibody? And so the challenge there for the actual circumstances is that you would really want to measure, whether that antibody is sitting in the muscle and is it really binding to myostatin. Now, again, similar to the drug circulation, it's really quite challenging to be able to look, inside each muscle to see if you have antibody in the muscle. So what we did again was, as a substitute, we looked at the blood to see if there was binding to that myostatin that was circulating. And so again, it's not really a direct measure. So for that reason, I gave us a little yellow check mark there, is because it's not really as good as being able to measure actually at the muscle. And then the third pillar that we look for is if there is a downstream pharmacologic reaction as a result of that binding or that inhibition. So typically what we would look for here is we want to know if there's a biomarker that changes to some degree that would be able to predict clinical efficacy. So is there something that's changing in the body that is going to be able to predict some kind of downstream functional change in these patients? And this is where, unfortunately, we get an X, because there really is no biomarker that's available to measure the pharmacologic change of an anti-myostatin drug. And with understanding how it's going to impact clinical efficacy. However, what we were able to do in our study is look at imaging as a potential biomarker to use for future studies. And my colleague Sarah is going to talk more about that. But that was one of the areas that we would hope to develop, is really understanding how we can use imaging as a biomarker to predict clinical efficacy. So the next slide. So briefly, this is the study design of our phase 2 study. We had 120 Duchenne boys who were 6 to 16 years old who were enrolled in a study that took place over 2 years. The first year of the study, after the first year of the study read out, we had a primary analysis. And for boys who completed through the second period, they went on to an open label extension. And this study took approximately 4 years to conduct. We went through the results of this trial, which are available on YouTube. I provided a link there, and I'm sure PPMD can provide other access to that webinar. But primarily, unfortunately, the efficacy was not demonstrated as being statistically significant over the difference between the active and the placebo by 49 weeks. However, we did learn that the drug was safe and well tolerated. We could measure the drug in the serum as we were hoping, and we did see that the binding actually did happen in the blood and serum as well. And if you go to the next slide. The other important things that we learned about were about glutamate dehydrogenase. So when we do drug development, one of the areas that drug developers typically want to look at is the liver. And the reason for this is because drugs are primarily metabolized in the liver. It's really important to be able to that during drug development, just to make sure that we don't have any toxicities. This is especially challenging in Duchenne patients because the markers that we use to look for toxicity in Duchenne patients, ALT and AST, not only come from the liver, but they also come when a person has injured muscle, such as Duchenne patients. So because they have these elevations in ALT and AST, it makes it really challenging to interpret if we have true drug toxicity due to the drug or if the elevations are actually due to muscle. So what's nice is that we have this glutamate dehydrogenase, which is a biomarker that's specific to the liver. And we were able to measure this biomarker in Duchenne patients and really establish that this can be used as a liver-specific injury biomarker. And we have now a large database of information with GLDH long-term with Duchenne boys that can be used as reference for future drug development, really in understanding toxicity related to drugs. Next slide. Troponins are a group of proteins that are found in both skeletal muscle and cardiac muscle, and they're responsible for regulation or contraction of that muscle. Cardiac troponin I is specific to the cardiac muscle, and when the heart is damaged, that protein is released into the bloodstream. Doctors typically use cardiac troponin I when they suspect somebody may have had a myocardial infarction or have damage to their heart. But what we've seen in Duchenne patients is that, the heart is also a muscle and it's injured. So therefore, this cardiac troponin I can actually be released into the bloodstream, even when there's not a myocardial infarction. making really interpretation of these findings quite challenging. So what we found in our trial was that the cardiac troponin I was actually elevated in about 20% of the patients, even at baseline before they received any drug. However, those elevations were not sustained over time, nor were they associated with clinical symptoms. This longitudinal data really has increased the understanding of how cardiac troponin I behaves in boys with Duchenne, and it really provides a reference data set for future clinical trials, especially where we're looking at drugs that may impact the heart as we study Duchenne. My colleague Sarah is now going to talk to you a little bit about imaging. If you can go to the next slide.
Great. Thank you, Shannon. So we had multiple MRI uses in this study. First, we had MRI to monitor for liver and cardiac safety. And then we also had a Psi MRI strategy to monitor for efficacy. We were very fortunate to be able to look at the body of work that Imaging DMD has put out with natural history studies, and that really supported the basis for us including MRI in our clinical trials. So they've done a great job of demonstrating that MRI correlates with disease progression, and that was part of our motivator to include MRI in this study. What we did is we used very similar methods that Imaging DMD has implemented, but we had a slightly different variation on the analysis. We used multiple MRI methods to characterize different disease processes. First, we had an imaging sequence to look at muscle volume across the entire thigh. We did T2 mapping, which it's a quantitative measure, but it can be used to evaluate muscle quality. And then we also did fat fraction imaging, and that was a quantitative measure of fat fraction across the entire thigh. So we wanted complete characterization of both volume and quality of muscle. And really what we learned is that these imaging biomarkers, they need to be scalable and widely available. During the site selection process, we could not let imaging alone drive site selection. So we needed to be able to bring the imaging to the clinical sites that had the experience with running these DMD studies. Next slide, please. So a little bit about how we implemented MRI in this study. The first thing that we learned was we really needed to work with patients to prepare them for the MRI imaging. We held sessions at investigator meetings to talk about strategies on how to prepare boys. And it was great when facilities had access to things like mock MRIs or child life specialists, but you can't count on that being present at all sites. So even something as simple as listening to YouTube videos of MRI scanner noises can be helpful in preparing a boy to go through the MRI scanning process. We were able to set up scanners at all sites in 31 sites in 8 countries. And as a reminder, these sites were selected based on their clinical capabilities, and then we would set up the imaging at the site. So we needed to use widely available scanning sequences. And it should be noted that while it helps for a boy to have access to video, it was not available at all sites. So all sites did have audio capabilities during the scanning session, but not necessarily video capabilities. We were pleased that all boys were able to undergo the MRI exams without sedation. There were cases where we had motion artifacts, and this was a common issue, and in some cases we had to drop those scans from our analysis. In order to harmonize across the different imaging sites, we needed consistent patient positioning and image acquisition protocols, and so we distributed equipment to sites. On the right-hand side, you see blue positioning pads that were available at all sites to help make the boy comfortable during scanning, and also get consistent scans across different imaging facilities. We learned quality control is going to be incredibly important in any future imaging study. Having site training and qualification processes in place. Phantom scanning is something that you can do on an ongoing basis to check that the scanner is functioning properly. And then we had ongoing image quality control checks. From an image analysis standpoint, we really need scalable tools. These images are very difficult to segment or analyze manually, so you need some sort of semi-automated tool. We used a central review process with study-trained radiologists. You need some sort of data quality monitoring process. So what we learned, it was a lot that went into the setup and execution of the imaging component of this study. We did learn that it is feasible to execute imaging in these larger studies. We still don't know the magnitude of change that's needed on an imaging endpoint to inform a future functional change, but that's analysis that's ongoing. And then this is, we learned that we could build a global strategy for imaging in DMD studies, and we are able to reuse some of the placebo data set from this study for future gene therapy studies. Next slide, please. And I'll hand it back to Shannon.
Thanks, Sarah. Finally, we just wanted to talk about sort of some of the reporting that's been done for the study, which we do provide all of our results on the clinicaltrials.gov website, as well as the clinical trials registry in the EU. However, if you've ever had an opportunity to look through those, you might find them not really comprehensible. In Europe, there is a plain language study result summary that has also been provided for the study, and Pfizer provides clinical study report synopsis on our website. And then the results of this study have been published as a primary publication with Dr. Wagner. We also have multiple other publications that are being worked on as we speak. In addition, we've had multiple opportunities to present this data at the MDA and PPMD, as well as Myology and the World Muscle Society. And then finally, we wanted to say that we have shared our placebo data with Critical Path Institute with the Duchenne Regulatory Science Consortium, and that was done in May. This is a group that is a collaborative research group that supports data sharing, and access can be provided to this placebo data upon request. Next slide. So finally, we just wanted to say thank you so much for all the boys and the families who committed their time to participate in these studies. We also are really grateful for the investigators and staff. Right now, we are in the process of working on publication of the results, and they really have been very enthusiastic about the data set that has been generated through this study. And then finally, we just want to thank PPMD and all the other advocacy groups for their support over the years in connecting us with many patients and families who've helped to really make this clinical trial a success. Thank you.
Thank you both. That was a great presentation. And just some housekeeping really quick before I throw a question back your guys' way. For anyone who's listening, if you have questions, please feel free to put them into that the little chat box down in the left-hand side on that little icon. We will take those questions when we get to the open Q&A. And I just wanted to kind of build a little bit on your comment from sharing the placebo data with de-risk. I mean, that's something that we love to see, and we know that's something data sharing the FDA has been very outspoken about the need for that. Could you provide a little more context in terms of what from the placebo data is being shared in terms of whether it's functional assessments, the biomarker data that you've collected, some of the imaging data?
Yep, so we have provided all of the functional data that we've collected, the lab data that we've collected, and some of the biomarker data is available there. I will say that not all of it is available because some of it we ran as exploratory, so it wasn't run in the same way under the same criteria that we ran the rest of the study. The imaging data is, the DEXA data is available there. We haven't yet transferred the MRI data, but that is coming.
Great, thank you. And then just one more question. So I really liked the approach that you shared about how you were tackling sites and MRIs, because as we all know, that can be a challenge in terms of availability and expertise. And I really like the approach that you're not letting your site selection be biased by who has that availability. Is that a strategy that Pfizer is going to continue to implement as you continue to design trials in Duchenne? And is that something that potentially some guidance for other sponsors in the space could be developed to help create a more equitable clinical trial landscape for patients who, their local sites may not have that expertise?
Yeah, I think it's going to be important that as we think about implementing these biomarkers at multiple sites, you need some base level of access to an MRI scanner, but they need to be widely available and easy for sites to implement. And so there may be a rigorous training component in getting a site set up to be able to acquire these scans, but we did learn that it's feasible to execute in these studies. And I think that's a strategy that we're going to take moving forward, that we need to be able to have imaging that can be implemented into an interventional trial without designing the trial around an imaging endpoint.
Great, thank you. And so next we're going to hear from some of the members of the Roche team, and they will be speaking about what they've learned from their trials with RG6206, which is also an inhibitor of the myostatin protein. And their trial was terminated in November of 2019, but as we learned with Pfizer, there's still a lot that can be learned here. So we're going to be joined by Dr. Roxana Degreci, the Global Development Lead of Microdystrophin, and Dr. Elena Zuravleva, who is the Patient Partnership Director. And so I will turn it over to the two of you.
Thank you, Eric. So I'm Roxana Degreci, and thank you again to PPMD for this opportunity to talk about our RG6206 lessons learned in Duchenne. Next slide, please. Our disclaimer is that this compound and its uses are investigational and have not been approved by the US FDA, and this information is presented only for purposes of providing the general overview of clinical trials and should not be construed as a recommendation for use of any product for unapproved uses. Next slide, please. So I just want to remind everyone a few things about RG6206, which is an investigational medicine for Duchenne. As we all know, patients with Duchenne experience a progressive muscle degeneration from their early childhood years. And as was mentioned at the beginning of the meeting, blocking myostatin has been shown previously in studies to increase muscle size. RG6206 is a fully human anti-myostatin adnecting that has been investigated in patients with Duchenne, and it blocks the ability of myostatin to restrict muscle growth. Next slide. This is a quick overview of the clinical studies up to date of RG6206 in Duchenne. So I just want to remind everyone that this is a molecule that was in-licensed by Roche from BMS. And when this was in-licensed 3.5 years ago, the phase 1 study was already completed, and the phase 1b/2 study was ongoing, and the phase 2/3 study was already designed and ready to start. So the study 0225 was a study of safety of RG6206 in healthy adults. And after that was completed, the study 40226 was started, and the main purpose of that study was the safety and tolerability of RG6206 in ambulatory boys with Duchenne, and that is known as the Thunderjet study. And after the results of that study, study 40227 was already underway, and it was already designed and planned and ready to start when we took it over. So there were a lot of activities where we still had an opportunity to put our stamp on, and then we executed that study up to November of last year, as Eric mentioned. And the main purpose of that study was safety, tolerability, and efficacy of RG606, again in ambulatory boys with Duchenne, and that was known as the Spitfire study. Next slide, please. So in the phase 1 study, which was the 40225 study, 103 adult volunteers have been exposed to RG6206, which was generally safe and well tolerated, and the free blood bound setting decreased as the dose of RG6206 increased. After multiple doses, increases in the size of thigh muscle and total lean body mass were seen. The Thunderjet study included 43 ambulatory boys with Duchenne, and RG6206 decreased free blood myostatin as it was expected, and imaging outcomes have suggested a positive effect on muscle mass and composition. And there were no clinically significant safety concerns identified. So the Spitfire study, which was the phase 2-3 study that we resumed in November of last year, has included 166 ambulatory boys. And the futility analysis of that study was conducted after 30% of the patients have completed the 48 weeks of treatment. This was a pre-specified futility threshold, and that was not met, and it was based on the North Star scale. Analysis of all the secondary and exploratory endpoints also did not indicate a meaningful treatment difference. Therefore, the program was terminated, which was both the Thunderjet and the Spitfire study. Next slide, please. So now going to the topic of today's discussions is what have we learned from this study. As it was very nice presented, practice makes perfect. So every one of these studies is an opportunity for us to learn, for all of us to move this forward. So as I mentioned, the program was in license from BMS, and the phase 1b/2 study was already well underway, and we couldn't influence it anymore, but we had the opportunity to look at the phase 2-3 study and make changes as we deemed appropriate. So the patients included in this study were 6 to 11 years old ambulatory, and they were randomized to receive either subcutaneous RG6206 or placebo on a weekly basis for 48 weeks. The number of patients included was 159, and they were randomized to one of the 3 arms. So 2 were active arms, and was based on the dose of 7.5 or 50 milligrams, which was based on the body weight of the patient, or 35 to 50 milligrams, again, according to body weight, or the matching placebo patients. The key inclusion criteria were genetic diagnosis of Duchenne, and all mutations were allowed. Patients aged 6 to 11, ambulatory, with a North Star score of over 15, and a 4-stair climb of under 8 seconds. Patients had to be over 15 kilograms and taking a stable regimen of corticosteroids. So one thing that we actually changed in this study after we took over the program was a change in primary endpoint. So when we took over the program, the primary was the 4-stair climb velocity. And after many discussions and a lot of analysis and close partnerships with the patient community and with all the senior leaders, we decided to change the primary endpoint to the change from baseline in North Star total score at week 48. And then the forced climb velocity became the main secondary endpoint, looking at the change from baseline at week 48. The other secondary endpoints were the traditional ones, the stand from supine, the 10-meter walk-run, the 6-minute walk distance, the POD-C transfers and basic mobility subscale, and the knee extension and knee flexion strength that measured by myometry. Another thing that I want to stop a little bit around is ActiMyo, which was our digital endpoint, and it's an exploratory endpoint that we looked at as change from baseline in lean body mass index at week 48 also. Also, we had a number of PROs that we looked at, and that data is also exploratory. Next slide. I will hand over to Elena to talk to you about the patient-centric outcome measures.
Thank you, Roxana, and thank you from my side to PPMD for inviting us to share our experience in this program. I think I really want to build on what Roxana been talking in the beginning, really, that in addition to the changes and adaptations in protocol, we've included several endpoints, secondary or exploratory, that were, to our and community's opinion, really patient-centric, and they've been used really to complement assessments in clinic, and then providing those unique insights relating to motor functioning. So one of them was stride velocity 95th percentile. That's been measured with ActiMyo. So ActiMyo is a wearable sensor that in our program been worn on ankles, and it records real-world data, ambulatory parameters with demonstrated accuracy. So among other parameters, we've been looking at the step velocity, stride length, as well as distance walked per hour. And its really unique features is that it objectively detects different gait parameters and ambulatory capacity on a truly continuous manner every time the boy's been walking or climbing stairs. And again, as I said, those parameters are measured in real-world settings, and they're independent from motivation or other confounding factors that we know are often a challenge in clinic assessments. Another endpoint that's been implemented as exploratory and been developed in close cooperation with the community is caregiver assessment. It's been recorded through an application installed on caregiver's smartphone, and the unique feature is, again, the possibility to film patients at home, for example, walking. And again, the changes that we were able to record and functional abilities were performed in a familiar environment. And those videos been reviewed and scored by expert physical therapists for changes in quality of movement. Ultimately, those endpoints, they really highlight the learnings from real-world data that are important, especially in the light of how heterogeneous Duchenne is. And if you think about our lessons learned, then on the next slide, it's really all about understanding and partnering with the community. And from our perspective, it's really truly acknowledging our partnerships, understanding of the community, and acknowledging their first-hand knowledge about the Duchenne. And as I said, that's been really true partners in this journey together. So as Roxana previously mentioned, we've been working together on the protocol development. We've been looking at the schedule of frequency of assessments. And ultimately, that's been all serving the purpose of reducing trial burden, looking at the ability of having remote assessments, minimizing travel needs for patients and for the families, as well as reducing procedures and site visits. And I think it also requires quite significant mindset change for us as scientists and drug developers, because we always want to get as much as possible from any type of clinical study. And that kind of research, it really makes us focus on the essential assessment must to have. And also, I guess another important point that that kind of changes in the protocol and partnership with communities, that really requires close interactions between drug developers and the communities and study participants. So we do have a common understanding of each other's must-to-have, if you will. And I guess I won't be original speaking now about the COVID impact on clinical trials and on the Duchenne community. So all those learnings that had and experienced during our program, it really becomes for us new default and new operating model in whatever forward-looking clinical programs we're talking about. And another very important learning is really selection of functional and meaningful endpoints for patients. We spoke before a bit about the North Star and digital endpoints, and it's really in rare diseases it's all about really moving away from those traditional endpoints or complementing them with those that are relevant or adapted to the particular indications we're talking about. So we've also developed clinician and caregiver impression of change measures that's been assessing meaningful changes in symptoms and abilities of voice with Duchenne, and those patient-reported outcomes been implemented as exploratory endpoints in the Spitfire program. They've been included in our clinical trials, and I think that's another example of complementing traditional endpoints with the novel ones.
Okay, and I would add one more thing, that obviously on this call, I think we are the second anti-myostatin molecule that unfortunately the program has been terminated in Duchenne. So that just goes to show, obviously, there's been others before us, before Pfizer and us, with programs with anti-myostatin in Duchenne. We know that the approach was not going to be disease modifying, and obviously the lack of efficacy has supported the termination of the program and the two trials that were ongoing. But that doesn't mean that it is a failure, like it was very nicely articulated in the beginning. And there's so much to learn, and we are still in the process of finalizing the clinical study report, and the data has not been fully published yet, but of course it's going to be. And we are working with the community on data sharing also, because that's the only way that we can advance is to learn from everything and put the data at good use to help everyone. So of course a lot of these learnings are going to be taken into our program, which is the collaboration with Sarepta on the microdystrophin gene therapy in Duchenne. Next slide. So of course, we'd like to take this opportunity to again thank all the boys and their families who have taken part in the clinical trials, as well as all our investigational site staff and the advocacy groups for their for taking this journey with us and helping us along the way, and of course for thanking the caregivers who provide a very valuable input into the design of clinical trials and all ongoing research. And again, thank you for the opportunity to be present here today.
Thank you both. And Roxana, I think you hit on a really important point there as you were kind of closing out. And something that can be challenging for the community, which is that, the data analysis does take time. And there is a bit of a lag from when, we have the announcement of termination of the trial before we start to see some of the analysis that we can learn from. And so it's important for us to keep in mind as we, try to maintain hope and keep our heads up. I did want to touch on the one point you were talking about some of your exploratory measures with remote assessment. And so one, if you could just kind of reiterate, where those two remote outcome assessments are in terms of development towards primary endpoints, that would be great. And then, is this something aside from, easing the burden in terms of minimizing visits, which especially in COVID-19 times is of utmost importance. Do you see this as a way to help get a more diverse trial population included into our clinical trial landscapes? That way, we have better information about how these compounds are going to broadly impact those living with Duchenne, since it is such a heterogeneous population.
Yes, thank you, Eric. That's a very good point, a very nice question that you're asking. And I always thought that the future is digital, and now more than ever, I think the future is digital and remote in the new reality that we all live in. So to go back to our two exploratory endpoints, I think on the XMI or the stride velocity, obviously that was a very important exploratory endpoint for us. And we're still analyzing the data, and there's going to be the data is going to be included in the primary study report, and we're also going to have a separate publication talking about this data. I think it's really important to have a novel way of having continuous monitoring at home that shows activities of daily life. It's not something that you do just at the clinic. So you have multiple data points, which is of great value. So this is one, I think, of the biggest learnings coming out of the program. And stride velocity, as some of you might know, is already an approved endpoint in Europe. So I think that's a great gain, and I think the field is moving towards that. And I think with the video assessments that Elena was talking about, unfortunately, the data collected there is much more limited. So we are working together with Casimir on how we can best analyze that data, since it's it's not the same quantity that we have with Actimayo, but it is something that we can definitely learn from, and anything that comes out of that will be made public for sure for the community to benefit from it.
Great, thank you. And then I think we're going to move on to our last speaker before we open it up for Q&A from the community. And so our final speaker today is going to be Wave Life Sciences, and they will be discussing their trials on their exon skipping product for those amenable to exon 51 skipping, suvodirsen. So this the clinical trial was terminated in December of 2019, and we are going to be joined today by Dr. Matthew Anderton, the Director of Clinical Operations, and Jeffrey Smith, the Senior Director and Head of Patient Advocacy.
Great, thanks very much, Eric. I'll kick it off. This is Jeff. First, I just want to say thank you to you and the team at PPMD for the invitation and for coordinating this session. I mean, it's obviously an important topic and not one that we often get together to speak about. So I just want to thank you and commend you and Pat and the team for identifying that and as always, leading us on these difficult but important conversations. So thank you for that. I also want to thank, everyone out there who's listening, all of you in the community who have supported our organization and our research and our colleagues, Pfizer and Roche and others over the years. Thank you for your support. Also want to, thank sponsors, researchers, clinicians, others who have supported and helped us, including, sponsors providing their data to us. And Matt will talk a little bit more about that. But, it truly is a remarkable community which has, really come together to advance research. And I think as we're learning today and as we're discussing today, we do have some key things that are helping to advance research in Duchenne despite disappointing, trial results. So if we can advance one slide, just to provide some background, as many will recall, Eric, and as Eric just mentioned, we were pursuing an exon skipping approach in Duchenne with a lead program called suvodirsen, which was intended to skip exon 51 and restore dystrophin production. So we ended up completing a phase 1 study and had an open label extension study that was ongoing when we received, biopsy results, which, proved disappointing, where we didn't see dystrophin restoration. And based on those results, we ended up discontinuing the program in December 2019, as Eric mentioned. But again, as I mentioned, and as we're talking about today, I think there are several learnings that we have that we took away that I think can lay a foundation for which future work can be built. And Matt and I will take you through a few of those, and I'll just turn it over to Matt to kick it off.
Okay, thanks ever so much, Jeff. I'd just like to reiterate what Jeff said and thank, both the patients and the families that took part in our clinical trials, but also the wider DMD community who's, we wouldn't be able to do this without the collective efforts in helping companies like Wave to be able to do clinical trials in DMD. So if we can just go to the next slide. So in terms of the topics of this webinar, we just first off like to touch on one of the aspects of which sort of I think highlights what we can learn from these studies. So one thing that was unique to the suvodirsen phase 2/3 trial was that it was selected as one of the first ever studies to be part of an FDA pilot program for complex and innovative trial designs. And the FDA's willingness to pilot this in DMD, I think, is testament to the strides that the whole collective DMD community has made in improving the standardization of clinical evaluations, both as part of clinical trials and also in observational studies and just routine practice at these DMD centers. So participation in this FDA program enabled Wave to have a number of interactions with the FDA in the design of the clinical trial. And whilst we didn't make it as far as we would have liked in conducting that trial before, as Jeff mentioned, we had the negative biopsy results, we hope that the design of this trial could be used as a model for future trials in Duchenne, as well as other rare diseases. So just to give you a bit more detail on this CID program, this complex and innovative design program, the selection of the trial into this was based on structures, and one key aspect of it was to be able to leverage DMD historical data from other trials and natural history studies to augment or support the placebo arm of the suvodirsen study. So through that, we intended to reduce the number of patients that we would need to be able to test the effect of the drug on the functional outcome measures of the study, which included things like the North Star ambulatory assessment. And in particular, minimize the number of placebo patients we would have required, and also potentially, accelerate to the completion of that study. Details of the design will be presented, at upcoming scientific meetings. And it's our hope, as I mentioned before, that this design can serve as a model for future DMD studies. And in the context of what you've heard from the previous presenters from Pfizer and Roche, regardless of a negative outcome for the drug being tested, the trials themselves still give us a huge amount of valuable data, and it wouldn't be possible to do complex and innovative design trials without the availability and sharing of historical data and learnings. And so that leads me on to the second point that we've put on this slide, which is given the dedication and commitment that the patients and their families made to the study, we felt it was only right not to only share the collective study results, but also to the individualized patient summary. So the individual patient results have been given to the principal investigators or study doctors so that they can use these as the basis to explain to the patients and their families the individual results. And we felt that this would be best shared through the investigators themselves, as they will be able to provide, further explanation and context to the results, and also answer any questions that the patients might have. So as well as obviously those individual results, we are, committed to sharing the collective results of the study. We've provided them to be posted on the UDRAT website, and we'll also share them with other efforts within the community as well to support future clinical trials. And to go through the last point, I'll hand it back over to Jeff.
Great. Thank you, Matt. So the last thing that we would highlight is, even more than a lesson learned, I think it's really just a recognition that engaging with you all out in the community, the Duchenne community, has truly shaped who we are as a company and who we are as individuals. We're not a big company. We're a relatively small biotech company and a young company. And more so than that, we're a collection of individuals who really just want to make a positive difference in people's lives. And, our engagement with you all inspired us, motivated us, energized us, and really cultivated that desire to make a difference. We've truly seen through listening to you all how important it is and to understand what you care about, what your needs are, and what your priorities are, so that we can incorporate that understanding into everything we do. I think we heard some of that from Roche, and I'm going to reiterate some of that as well. One of the first things we did early on was to talk to individuals and advocacy organizations like PPMD. I'm sure they'll recall meeting with us early on to discuss our research and our clinical trials. And we ended up changing several aspects of the studies to address the needs of those in the community. The CID program that Matt just spoke about is one example that arose out of feedback that we received early on in discussing the needs and priorities of those and opportunities that we could use to reduce the burden of clinical trial participation on individuals and families. Another example is our work to minimize the burden of biopsies. We know how challenging those can be to families, and so we worked to reduce both the number of biopsies and to choose a location, where we took the biopsy that would still provide the tissue quality that's needed to perform the analysis, but minimize the visibility of the scar. The boys who are participating in our studies have to live with the scar in many cases for years. So we wanted to take that feedback, and we did take that feedback and incorporate it, and ended up taking it from the deltoid, the shoulder muscle, where it would, the scar, if there were a scar, would be less visible to others. There are other examples that we took. I think the takeaway there is that, it was just immensely valuable to us, and I think definitely could serve as a model and will serve as a model for other studies and for other sponsors as, research progresses, to make sure that you're understanding what's meaningful to individuals, to families. You're incorporating that into the study so that you're measuring what's meaningful, and you're reducing the burden as much as possible, so that you can ensure that you're getting a rigorous study, but one that is able to get a clear scientific answer in a way that's that's meaningful to individuals and families that are living with these challenging conditions like Duchenne. So another thing that we did was, in terms of community engagement, was to bring parents together to talk about the lived experience of families. And the objective of that work was to, get a depth of understanding of the needs and priorities of families, so that we can incorporate all of that into the activities that we lead, activities and initiatives, and consider and develop potential solutions that can support families. Ended up having 3 meetings, which brought together 30 parents with us, ultimately, representing 35 boys and 1 girl with Duchenne, all of various ages. And, not only did we learn a ton about what it's like for a family living with Duchenne, but it was also an inspiring experience for many of us, and I'm sure some of the parents as well. At one point, we had a group over to our offices in Cambridge for just a brief reception, and so they were able to meet some of us and some of our employees and see our labs. And the next day, a few of our scientists showed a few of us Post-it notes that had been left on their computers by some of the parents who had joined us at the reception. And they were just like brief notes of thanks and of inspiration, and just notes of talking about what it meant to them to have these researchers staying and working on behalf of those of their company and of those in the community. And our folks were really, really touched by that and continue to be touched by that and still talk about that. And that's just, an example of how, again, engaging with you out there has shaped us both as a company and as individuals. We've continued to, incorporate things like that into our work going forward, even though our work in Duchenne has not continued at this moment. The legacy of that work and the learnings of that work definitely lives on. So, again, I just want to thank all of you for that. If we can advance to the last slide, and for everything the support over the years. And Eric, happy to answer any questions.
Oh, thank you both. And that I think that's a really kind of powerful message about that interaction between yourself and the community. And I think that's, important and great to see and hear how you are incorporating the patient voice. And also great to see that, for those researchers and those kind of not living with Duchenne day to day, are able to kind of connect on another level, and use that as kind of inspiration for the work they do. Also, we're very excited about the complex innovative trial design. Although we didn't get to see it kind of play out, we're very happy to see that information about that is going to be shared. Could you comment on in terms of the placebo data you were going to be comparing to the other sponsors from the Duchenne space that were contributing data, just in kind of showing some of that work that was being done between the different groups?
Certainly. I mean, I think in terms of the data sets, there were multiple, and I think, it's from a number of different organizations, and all those who have done research in the DMD space and conducted trials. So it's, we had Prosenza, GSK, there was some data from Pfizer, PTC, so, and also historical data collected by groups in Italy and by data sets in Leuven in Belgium. So I'll have missed a number of them, but yeah, there was an awful lot that was part of that historical control data.
Great. It's wonderful to see kind of that kind of interaction between the different groups. And then if we could just go to the next slide before we open it up to questions from the community, and we have a few that have come in already. Just to kind of further on Jeff's point about, listening to the patients and taking that patient perspective to heart. we do have a survey for those in the community to help better understand your experience with trial termination. So if you've participated in a trial that was stopped or terminated, we are asking to learn a little bit about your experience at join.parentprojectmd.org/trial story. This is something where we can take your feedback and again make that something we share with sponsors so we can learn about better ways to communicate something that can be a difficult experience. And just as a note, one of the questions came in to clarify. So when we're talking about termination or stopping of study, what I mean by that is a study that is stopped early, that is not going to start again, and then the participants, so those participating in clinical trial are not being examined or treated. Whereas if you look at a trial that's completed, that is just one that has made it to the end of its kind of normal scheduled visits, where the last patient in that study has completed their visit. So just a little bit of a difference here. We're talking about trials that have stopped early. And with that, I think we are going to open it up to questions. So Pat, as well, if you want to hop on. The first question that we had, and this one is directed to Pfizer. Could you comment on what role the imaging DMD study played in assessing your MRI outcomes? The imaging DMD study, for those who don't know, has been a 10-year-long natural history study of imaging with a number of institutes and lead PIs down at the University of Florida.
Sure. So imaging DMD has really put a lot of information out there to demonstrate the value of incorporating MRI-based endpoints. A lot of their work has focused on fat fraction and T2 mapping protocols, and while we did not use the exact same implementation or analysis methods, we did go after those similar MRI techniques of incorporating both fat fraction and T2 mapping. So their work provided a lot of guidance for us to develop protocols that we could scale up and execute as part of our clinical study.
Maybe I could just also add that when we were initially developing the imaging portion for the study, they were also tremendously helpful in helping us think through some of those considerations. So they were very collaborative, in their help with developing our program and our considerations.
Oh, great. Thank you. I think that's good to know too, that there's the standardization of some of this data collection as we go across. I think that's really important when we then go to look broadly at trying to bring together data sets that way we can, build better outcome measures. So great to hear. Now this would be a question kind of for all three sponsors. Was there any learnings that you could extrapolate to the non-ambulatory populations, or your development of non-ambulatory trials from these studies that were all kind of based in a younger ambulatory population? And I'll just maybe start with the Roche team and jump around.
Thanks, Eric. Well, of course, the study has been focusing primarily on ambulatory population. But I think as we've been discussing it also earlier, use of digital endpoints is something that is reality now and getting more and more incorporated in all the programs moving forward. So while initial, I think, steps were done again for the ambulatory population, those endpoints or those technologies can and should be definitely used in the non-ambulatory population as well. And especially if we are talking about home assessments, video assessments, right? So again, while it's been originally used in the Spitfire study, I think for all sponsors, it's something that we're all considering this, incorporating in all our future programs.
Thank you. And with Pfizer, I mean, does some of the work you've done with the MRI imaging and looking at glutamate dehydrogenase, can all of that be kind of as well carried over into a non-ambulatory population if you were working with that group?
Yeah, this is Shannon. I would say yes, absolutely. Our learnings from GLDH and also the cardiac troponin I are really important, and they are enabling us to look at an non-ambulatory population now. Sarah, do you want to talk about the imaging?
Yeah, I think that's going to be something that we do start to move forward is looking into implementing some of these techniques in a non-ambulatory population.
Great, thank you. And then a question that came in actually for Wave. With the data that was contributed from the different pharma partners that you were working with, was this data all integrated, or were they utilized as kind of individual data sets for your trial design?
So the plan was obviously to, as we got further along, was to integrate them all together, but they were used as individual data sets. And, you know, through those, because there is a huge amount of data, particularly in some of the natural history studies, we had to then be able to select the patients that closely matched the population that would have been in our phase 2/3 trial. So, you know, you did need to drill down into the individual data almost to the patient level to be able to supplement our study.
Great, thank you.
Thanks for that. So just a quick question, it's Pat, Matthew, and Jeff. So did you do an individual patient match, age, weight, mutation, and using individual data sets?
Well, the mutation wasn't always available for each individual patient, but where there was that data, then yes, that was part of all the statistical analysis that we would go to that level to try and match patients as closely as possible to those within the study. So that's why you need the joined up data and as much data as you can get, really, to be able to fine tune that.
Yeah, thank you for that. That's very great. Thanks, I appreciate it.
So we had a question come in, and I think it's maybe building a little bit off of some of the comments from Wave in terms of how you shared data back with the individual patients, which is great to see. So for kind of all of the sponsors here, could you maybe explain the difference between how the data that might go back to patients versus data that might be shared kind of with other sponsors or more broadly in the community, and what that looks like in terms of how it's presented through either publications or kind of agreements with someone like D-Risk? Just kind of the difference between the patient level data and kind of the aggregate data from your studies.
Yeah, so
maybe
go ahead. Sorry, go ahead, Eric.
Oh, no, you're fine, Matthew.
Okay. So, obviously the aggregate data, you tend to have mean and median average results displayed and a standard deviation around that. What we did with the individual patient data is, it is just that. It was all their functional assessments, it was summary of all their safety, the key laboratory bloods that were taken, even some of the images from the biopsy. So we had, immunofluorescence for the individual patients, so they could see what their muscle fibers look like at the start of the study and what they look like at the second biopsy, post-treatment. And so, it was really just to be able to showcase that to the families and through their investigators. So if they wanted to see it, they could ask their investigator for it, and the investigator could talk through it.
Great. And then with Roche and Pfizer, is there plans in place as well to share that patient-level data back with the individuals who participated in your study through, their clinicians?
This is Shanna from Pfizer. Yes, we have shared the individual patient-level data back with investigators. That's actually part of the standard practice that happens at the end of every clinical trial. Each of the investigators do have that data available to them. And I guess just you had asked a question about the differences in exactly what's shared. So when we share this Critical Path Institute for D-Risk, that's actually raw data that's shared from the placebo data patient set, and then the analyses that we run, those are what ends up in publications. I'm not sure if that was what your question was, just in terms of what's specifically shared. And then they can respond.
Yeah, no, that was. Thank you.
And Shannon, if I may, I'm sure that all of you have experienced where perhaps as individual data is returned, or even prior to that, where the patient, and Jody Wolf highlighted this, but it's always on all of our mind, and the patient or the individual patient says to you that my, in this case, son, was doing better on this particular drug. And how do you respond to that in such a way? I mean, we know there's a placebo effect, and we hear it and hear it and hear it, but at the same time, we can't really answer that question, right? Because you're looking at statistical analysis. But how do you manage that?
Yeah, I would just say that yes, exactly to your point, Pat. We definitely heard back when we announced the termination of the domabozumab program that some of the families felt that they did better. And actually, that's often the case when people participate in clinical trials. For whatever reason that is, placebo effect, having more contact, and oversight, for their disease, having more interaction. But unfortunately, the way that study is set up, as I said earlier, is that we are testing a hypothetical effect around a drug, a statistical test that, we need to carry out to the analysis to show whether that, hypothesis can actually be true or if it's not proven. So yes, I think it's actually pretty common in clinical trials that people do feel better when they're participating on a clinical trial, but unfortunately that doesn't always represent what the results are of the trial.
No, you're right. It's just a hard message to deliver, right?
Yes.
There's all sorts of reasons why you might feel better during a trial. I think right now in the midst of the pandemic, just seeing another person, I might feel better. So I think that, and I don't mean to make light of it, I just think it's such a struggle for all of us who watch these young men and may see differences in them. And I appreciate that statistically you're looking, but at the same time, it's a hard message, right? It's a hard message to deliver and a hard one to accept.
I would also say that one of the things that we heard repeatedly is about the terrific relationship that these families developed, with the investigational sites, the physical therapists, the study coordinators. And, you can imagine a family who'd, been on a trial for 2 years, 3 years, 4 years, that now we're saying this relationship is ending. That's also somewhat heartbreaking for families who have been, involved for so long and have developed these really close relationships, with these healthcare providers. So, I think that's also really, challenging and, difficult to hear.
No, you're right. And I'll get back to one of my favorite subjects, which is language. So if I had my son, if I was in any trial and I took my son, or in my case, sons, to the clinic, and the PI would say, "Oh, they look great," I would, in my head, it would be, "Oh, they're responding." Do you know what I'm saying? So language really matters, and how we deal with this is very sensitive and hard. So thank you.
Mm-hmm.
If I may just add to that, this is Roxana from Roche. We are a bit behind Pfizer, of course, because our program was terminated just in November of last year. But we are also in discussions with D-Risk for sharing data. Also, we're looking at potential online sharing platforms where then different groups can go in and pull the data. And also, of course, the patients can go back to the investigational site for their individual patient data, which is key for these patients.
Yeah, Roxanne, it's wonderful. I certainly applaud all of your efforts to return individual data. I think, again, trials are really burdensome to participate in. They are filled with anxiety, and we all want them to do well. And certainly when we're disappointed, that's hard. Those are very difficult. But I think the care you have taken, all of you online today, Pfizer, Roche, and Wave, have taken such care of the families to go back to them and really wrap your arms around them and help them understand. And I appreciate that, and I know this community does. Back to you, Eric. Sorry for the interruption.
No, you're fine, Pat. We just had one other question, and this is for both Pfizer and Roche. Kind of reflecting on that, we have had other trials in this space that have struggled with recruitment. Can you comment maybe on, was enrollment a challenge for your trials? And if not, what do you think kind of drove that success in recruitment? And is that, again, something that we can build upon as a community as we have this vast pipeline of trials that are becoming available? So maybe, Roxanne, I'll start with you.
Yeah, thank you, Eric. I was going to go anyhow, so thanks for giving me the chance. So actually, this was not a challenge for us for the Spitfire study. We did encounter some challenges that were not related at all to patient availability or desire to enter the trial. They were more drug related, so we had to stop screening during the first part of the study because of the transition of the program from BMS to Roche. But then right after that, screening resumed and actually completed ahead of time. So we didn't encounter any challenges. But in my previous life before Roche, I've also seen the other side of the story. And I think what I can say is it is a very busy space, and we all know that, and that is good and bad. So it's good for the community and for the patients and for the families because there are many things going on and there's a lot of opportunities, but it's also bad because there are trials recruiting at the same time, so there's a lot of competition, and of course, this is a rare disease, so there's only that many patients to go around. So I think it's still good, it's a busy space compared to other spaces, but obviously I think patients in this community are very well informed, and the design of the protocol, the molecules, mode of action, the burden of the trial, all these are factors that these families take into account, especially in the new world we live in. So we all need to be very cognizant of how we design our protocols moving forward and keeping that fine line between getting the data that we think we need versus expose patients to too many visits and unwanted risks. So I think the world has changed before our eyes, and we're never going to be able to do all the things that we used to, which is in a way a good thing, I think, for the burden for the patients.
Reducing that burden is pretty key. And I will say, I think it was really promising that in all the talks we heard from sponsors at the virtual conference, kind of that age range of that, 4 to 7, 4 to 10, we heard a lot broader inclusion being included, or trials aimed at non-ambulatory patients that we have previously, and a few in the Becker space. So it's always a bit more exciting that families that maybe before felt like once they lost ambulation they were unable to participate, now will have some more options available. And then Pfizer as well, if you would like to comment on enrollment for your trials.
Yeah, I would say we definitely had a lot of challenges around enrollment for our study. It took about 2 years to fully enroll our study. And I would just say that at the time, we were enrolling, there were a lot of compounds being considered and a lot of clinical trials ongoing. But I do want to commend PPMD and other advocate groups who really helped us, with the communication on what the trial was. And I think, one of the things that I heard throughout is that, families had a lot of choices. And I have to say, sitting in their shoes, I often wondered, how did they, make a choice between one trial or the next, or what was a good fit for them. So it definitely was a challenge, but obviously we did fully enroll the study eventually, and I really thank the PPMD and the other advocate groups for helping to enable that.
Well, thank you. I mean, that is something that we are always happy to advocate for. And I think we are just about at time, so I would like to thank, again, all of our speakers for coming in and talking today on a subject that can be, again, difficult. Trial termination is always a challenge, but I think as we saw today that there's a lot still to be learned in these instances, and it can continue to advance the field and provide benefit to all of those families living with Duchenne and Becker. And again, we appreciate all those families that are willing to participate in clinical trials and help us find answers and treatment for this disease. So as a reminder, this webinar will be recorded and posted on the website. And for those who, again, have participated in a clinical trial that has terminated, please look for that trial story survey. We would love to get your feedback and hear more about your experience. So thank you, everyone.
Thank you.
Thank you.
Thank you.
Thanks a
Thank you
lot.
all.
Thank you.